Prudent Scholars in association with Goa Headmasters Association.
Hello everyone. Today we are going to start with the new lesson from the science, and the name of the lesson is lesson number 6, life processes. Life processes, this is nothing but the basic functions that is performed by living organisms to survive on this earth. Some of the examples of life processes are growth, which takes place, then reproduction, nutrition, transportation, excretion. These are some of the examples of life processes. And in today's class, we are going to cover the life processes, one of the life processes, that is nutrition. So, this is lesson number 6 from our science textbook, and this lesson is related to biology, which is nothing but study of living organisms. So, we are going to cover in this lesson 4 life processes, that is nutrition, then respiration, transportation and excretion. Just now I told you regarding the life processes. Nutrition, this is nothing but the process of taking food in and its utilization by body. Respiration, this is the process in which organisms take in food and produces energy. So, during the process of respiration, using food, energy is released. Transportation, this is the process wherein the materials that are observed or made in one part of the body is transferred to the other part of the body. And excretion, this is the process of removal of harmful metabolic waste from our body. So, these are the 4 main processes that we are going to learn. In this slide, we can see the definitions of life processes, nutrition, respiration, transportation and excretion. So, we are going to start with nutrition. Now, autotrophs. In the previous classes, we are familiar with this word autotrophs. These are the organisms which prepare their own food by using simple inorganic substances like carbon dioxide, water in the presence of sunlight. Now, some of the examples of autotrophs are green plants which can prepare their own food, some bacteria, and we can see the image of one of the bacteria. Then we have something called as the heterotrophs. I repeat, we have something called as the heterotrophs. These are the organisms which never prepare its own food, but they are dependent on other organisms. Some of the examples of heterotrophs are animals, fungi. Now, we are going to cover regarding the autotrophic nutrition. Now, what is the meaning of autotrophic nutrition? It is the process in which organisms prepare their own food by using simple inorganic substances like carbon dioxide, water in the presence of sunlight. These organisms also possess chlorophyll, which is nothing but the green pigment. Now, we already know regarding the plants, and we have seen examples of autotrophs. The organism in which mode of nutrition is autotrophic, we call them as autotrophs also. One of the examples is green plants, and we can see here plant. They prepare their own food, they prepare carbohydrates, and unutilized carbohydrates in plants, this is stored as starch. In case of human beings, unutilized energy is stored as glycogen. Now, we are going to understand the events that occur during the photosynthesis. Photosynthesis, this is the process by which plants, green plants prepare their own food using simple inorganic substances like carbon dioxide, water, and they convert it into stored form of energy in the presence of sunlight, and these plants must possess chlorophyll, which is green pigment. As we can see the equation here, in the reactant side we have carbon dioxide and water, in the product side we have glucose and oxygen. This is the equation that represents photosynthesis process. In this equation there are two reactants, carbon dioxide and water, two products, glucose and oxygen. So, question may be asked, which are the two products that are formed during photosynthesis? So, these are the two products, glucose and oxygen. AQ stands for aqueous, L stands for liquid, and again L stands for liquid. Now, let us understand the events that occur during the photosynthesis. There are three events that occur during the photosynthesis. One of the events is absorption of light energy by the chlorophyll. As we know that green plants consist of green pigment called as the chlorophyll. So, what is done is light energy is absorbed by the chlorophyll. So, one of the events that takes place is absorption of light energy by chlorophyll. One more event that takes place, that is light energy which is absorbed, that light energy gets converted into chemical energy, and water molecules splits into hydrogen and oxygen. We have covered the second event that occurred during photosynthesis, that is conversion of light energy into chemical energy and splitting of water molecules into hydrogen and oxygen. One more event that occurs during the photosynthesis, carbon dioxide. This carbon dioxide gains hydrogen. I repeat, carbon dioxide gains hydrogen, and this carbon dioxide gets reduced to glucose or carbohydrates. I repeat, reduction of carbon dioxide into carbohydrates. So, these are the three events that occur during the process of photosynthesis, and we can see all the three events here, the statements we can see here. Next. Now, all these events may not occur simultaneously, or all these events may not occur one after the other. One of the examples to understand this is of desert plants which are found in desert. So, what desert plants, what they do? They take carbon dioxide at night and they prepare an intermediate food. They will not prepare the final thing, but they will prepare the intermediate food, and on the next day when the chlorophyll absorbs sunlight, after that, after absorbing sunlight, they prepare the food. In desert plants, all the events will not occur one after the other. Okay? We have seen the example in case of desert plants just now. Now, let us understand the cross section of a leaf. We can see this mid rib which provides the structural strength to the leaf. Then if we cut this, if we see the cross sectional view of the leaf, then we can see here two different types of tissue. One is phloem and the other one is xylem. These are also known as the vascular tissues, and we have learnt in 9th standard regarding this vascular tissues. Now, phloem is tissue which is present in plants which transport soluble products of photosynthesis. So, during the process of photosynthesis we have seen the products glucose and oxygen, right. Xylem, this is one more tissue that is involved in the transportation of water. So, one of the points of difference between the phloem and xylem is phloem transport soluble products of photosynthesis, xylem transport water in plants. Then if we see the cross sectional view of this part, okay, we can see here, then there is waxy cuticle. Now, this prevents, it has its function, it prevents the loss of water, it never allows the leaf to lose water, okay, to lose more water. Then we have the upper epidermis. This upper epidermis, it transfer most of the, or it allows most of the light to pass. Green pigment, green dots we can see, not the pigment. So, green dots, this is known as the chloroplast. Then there are air spaces where air can enter. Then we have the lower epidermis, and lower epidermis consists of many openings. Okay. We call the pores present on the leaves as stomata. Stomata. This diagram represents open and closed stomatal pore. The first diagram represents open stomatal pore and this diagram represents closed stomatal pore. Now, these are the pore. This is one pore, this is another pore. Now, this stomatal pore is open and this stomatal pore is closed. And around this stomatal pore, you can see this. These are guard cells. Now, guard cells has its own function. Guard cells helps in opening and closing of stomatal pore. So, guard cells open stomatal pore, guard cells closes stomatal pore. This is the function of guard cells. Then stomatal pore. Stomatal pore has its own functions. Normally, two basic functions performed by stomatal pore. So, stomatal pore helps in exchange of gases. We already know that plants require carbon dioxide and during the process of photosynthesis they give out oxygen. So, stomata helps in exchange of gases. So, massive amount of exchange of gases takes place through stomata. One more function performed by stomata is, we already know that plants lose water and the loss of water by the plants through the pores in the form of water vapor we call as transpiration. So, loss of water takes place through stomatal pore. These are the two functions performed by stomata. So, we have seen this functions of stomata and function of guard cells. This diagram can be asked in the examination. Now, let us understand two activities. One of the activities is, in this activity we will learn that chlorophyll is essential for photosynthesis. So, what we have to do? We have to take the potted plant having variegated leaves. Variegated leaves means the leaf having or the leaves having different color patches, especially green and white. Such plant is money plant, or we can take the croton plant. We have to keep this plant in the dark room for 3 days. Now, once we keep this plant in the dark room for 3 days, this plant will utilize the starch which is stored already in this plant. After 3 days, we have to keep this potted plant in the sunlight for 6 hours so that this plant can prepare its own food. After this, we have to pluck a leaf and we have to observe the leaf. there will be green color patches and there will be white color patches. So, we can make use of the tracing paper. Tracing paper must be kept on the leaf and we can draw the rough diagram of that leaf by keeping tracing paper on the top of the leaf, and we can mark the shaded areas, especially green color. Now, once we do this, keep the tracing paper aside, make use of that leaf, make use of this arrangement wherein we are going to make use of water in a beaker, stand, wire gauze, burner. We must heat this beaker and when we observe the boiling of water, when water boils, at that time we have to introduce that leaf in this beaker. Now, why leaf is kept in water which is boiling? Once we keep this leaf in water, boiling water, the cell membrane of the leaf, it breaks. At the same time, cuticle, we have seen regarding the cuticle, it becomes soft. Okay. Next, we have to remove the leaf and we have to keep that leaf in alcohol. In this case, we have to prepare the water bath. Now, water bath, as you can see here, in this beaker already water is present and that leaf now we have placed in alcohol, that is in test contains alcohol, and we have to heat the water till alcohol starts boiling. Okay. Once alcohol starts boiling, we have to keep the leaf for some time and we are going to observe that the green color leaf, that leaf will lose its green color. So, leaf will become pale, it will become dull. Now, why it will become dull? Because when we heat this alcohol, when alcohol starts boiling, alcohol will remove the green color pigment which is nothing but chlorophyll. So, at the end of this part of the activity, the color of this becomes green because leaf will lose its chlorophyll. We are understanding this activity that chlorophyll is essential for photosynthesis. So, after this we have to remove the leaf and we have to make use of the iodine solution. Now, iodine solution is used to detect the presence of starch and we have to keep the leaf in iodine solution. So, iodine solution can be taken in one of the test tubes, dip the leaf in iodine solution, after some time remove the leaf, rinse off the iodine solution and only the green color patches of the leaf, it will turn blue-black. So, these are the steps. As we can see here, this is the original leaf which was having the green color patches, only that part turn blue-black here. So, this indicates that the part which was green in color, that part only produced food. So, with this activity, we understand that chlorophyll is essential for photosynthesis. So, we have seen this activity wherein chlorophyll is essential for photosynthesis. Let us learn now carbon dioxide is essential for photosynthesis, as everyone knows. So, next activity is activity to understand that carbon dioxide is essential for photosynthesis. Now, in this activity what we have to do? We require 2 potted plants. Now, these potted plants may not be having the variegated leaves. Okay. So, we have to take 2 potted plants. Again we have to do the same thing. We have to keep both the potted plants in the dark room so that it utilizes the starch which is present. So, we have to keep these potted plants for 3 days in the dark room. So, starch will get utilized, these plants will utilize the starch which is stored. Next, after 3 days, we have to remove the plants and we have to keep those plants under the sunlight. Now, before keeping the plants under the sunlight, we have to do some arrangement. So, what we have to do? We have to take 2 plates, 2 plate घेवपाच्यो, and on each plate we have to keep the potted plants. At the side of one potted plant, we have to make use of the watch glass in which potassium hydroxide is present. Potassium hydroxide is an example of base. So, we have to make use of the potassium hydroxide and we have to cover both the potted plants separately with the help of jar, with the help of bell jar. This potassium hydroxide is used for one purpose. This will absorb whatever carbon dioxide which is present in this air. This will be absorbed by the potassium hydroxide. Here also air is present. Now, we can make use of one thing, we can make use of the Vaseline. So, if we use Vaseline here, then air will not be able to come out here in both cases. So, what we have done in short, we have taken 2 potted plants, we kept them in dark room for 3 days, they have utilized their starch. After 3 days, we kept them under the sunlight for 2 hours, we can keep them under the sunlight. And before keeping the sunlight, we must make this arrangement. Okay. Now, so after 2 hours, we can remove these jars. We can pluck one leaf, say from this potted plant A and potted plant B. So, keep in mind potted plant A is not going to get carbon dioxide here, and here carbon dioxide is available to this plant. And we have to follow the procedure that we followed few minutes back to detect the presence of starch. So, we will come to know which plant has produced food by doing the starch test, and to do the starch test we make use of the iodine solution. So, we have to follow the procedure now. We have 2 leaves now, leaf A and leaf B. Just now I said that we have to do the starch test. So, what must be done? We have to make use of the beaker. So, 2 beakers we have to make use of. We must pour water in both the beakers separately. We have to heat the beaker till the water starts boiling. We have to keep on heating. A stage will be reached wherein water will start boiling. And when water boils, at that time we have to introduce these leaves separately in the 2 beakers. So, once we do that, boiling, as we already know, when we introduce leaves in boiling water, at that time the cuticle becomes soft and cell membrane breaks. So, what will happen if the cell membrane breaks? When we are going to make use of the iodine solution to detect for the presence of starch, at that time iodine solution can reach to the starch. So, we have to introduce both the leaves in 2 different beakers. In both the beakers, water will start boiling, a stage will be reached wherein water will start boiling. So, we have to introduce both the leaves separately into separate beakers. A stage will be reached wherein, as we already know, what will happen in the leaves? Cell membrane will break and cuticle becomes soft, will become soft. We have to remove both the leaves and introduce two leaves separately in alcohol, and we have to prepare the water bath. Now, water bath, we have to take the container. For example, we can take the beaker, pour water, and we have to make use of the test tube in which alcohol is present, and in that test tube having alcohol, in that test tube we must dip this leaves separately. We have to keep the leaves in alcohol till the alcohol starts boiling. And after that, we already know that boiling of alcohol, when alcohol boils and if leaves are present, then a stage will be reached wherein leaves will become pale. Why they will become pale? Because leaves will lose its green pigment, and alcohol, it will become green in color because of the chlorophyll. After removing both the leaves from the alcohol, we have to introduce two leaves separately in iodine solution. Keep it for some time, remove both the leaves, and we are going to observe that leaf B or leaf B will turn blue-black. So, if leaf B turns blue-black, we understand that leaf B has produced food or starch is present in leaf B, whereas we will not see any change here in case of leaf A. So, leaf A here, this leaf A will not prepare its food because of carbon dioxide which is not available to this. Okay. So, these are the steps that we have followed to understand this activity, and based on this activity we conclude that carbon dioxide is necessary for the photosynthesis process to take place. So, we have seen two activities wherein chlorophyll is essential for photosynthesis and carbon dioxide is also essential for photosynthesis. We already know that carbon dioxide is absorbed or taken by the plants from the air, but plants also require other substances like other materials like nitrogen, phosphorus, iron and magnesium. Nitrogen is required by the plants for synthesis of proteins. Nitrogen is taken by the plants in two ways, in the form of inorganic compounds such as nitrates and nitrites, and in the form of organic compounds which are produced by the rhizobium bacteria which lives in the root nodules of the leguminous plants. So, they convert, they produce one compound in which nitrogen is present, and that compound is absorbed by the plants, and in this way they get nitrogen. They also require phosphorus, plants also require iron, and they also require magnesium. In this session, what we have learnt? We have learnt regarding the autotrophs, autotrophic nutrition, events that occur during photosynthesis, functions of stomata, function of guard cells. We have also seen cross section of leaf. Then activities, two activities we have learnt to detect the presence of starch, and based on two activities we already came to know that chlorophyll is essential for photosynthesis to take place, and carbon dioxide is also essential for photosynthesis to take place. So, these are some of the questions. Autotrophic nutrition, heterotrophic nutrition. It is the mode of nutrition in which organisms makes its own food from the simple inorganic materials like carbon dioxide and water present in the surroundings with the help of sunlight energy. Heterotrophic nutrition, it is the mode of nutrition in which organisms obtain their food from other organisms. Give one word for the following: the process of transferring a source of energy from outside the body of the organism, which we call food, to the inside. We already know this is nutrition. The process of acquiring oxygen from outside the body and to use it in the process of breakdown of food sources for cellular needs. Respiration. The biological process involved in removal of harmful metabolic waste from the body. Excretion. Photosynthesis. Then the products formed during photosynthesis. The three events that occur during photosynthesis. How does the process of photosynthesis in desert plants differ from those in other regions? These are the things that we have covered. Thank you.